Hi everyone, I'm going to talk to you about lessons. I'm going to talk to you about the genomic epidemiology work that we did for SARS-CoV-2 and basically the lessons sort of going forward. But actually, I'm going to divide the talk into two parts. One, it's going to be about when you have data, uh, how can you use it? And the second part is going to be when you have samples, that is when you have a surveillance network already set up and you have access to those samples, how can you work with that? Okay, so both of them are sort of positive success stories in a sense, and they are meant to highlight the challenges we have when we don't have these. So the work that I'm going to talk to you about has been done by a lot of people. Most of the sequencing work has been done by Pramada. And I'm going to talk to you. So these are all the people from NIMHANS, which is where I was as an early career fellow. A lot of the work that I'm going to talk to you the data was generated by other labs. So a lot of the INSACOG labs as well as InSTEM, that's for SARS-CoV-2. And I'm also going to talk parvovirus, and for that, all of the work was done at NIMHANS. I'll try and mention that as we speak. So as funded by the India Alliance DBT Wellcome Trust, we also had funding from DBT as part of INSACOG, which is a consortium for doing genomic sequencing in India. And we had some funding from the government of Karnataka to look for variants. And I believe in this sort of model where we need to understand a pathogen in the context of its host and in the sort of epidemiological context that it is placed in. And this seems pretty obvious, but to bring all these people together to work together, we need good supporting structures. And that's why I sort of started IDRF, which is Infectious Disease Research Foundation. All right, so I'm going to talk to you about the introduction and spread of SARS-CoV-2 in the state of Karnataka, a story of what is possible when we have data, early detection and introduction of variants of concern. I'm going to talk about alpha and omicron. Then identification, can we identify locally emerging variants? Where are we with respect to that? And then environmental surveillance for SARS-CoV-2. Once I complete that, my plan is to take you through a story about a parvovirus in West Bengal, which is associated with acute meningoencephalitis. All right, so this is basically the first 1,500 cases looking at March to May 2020, very early on in the pandemic. These are the first 1,500 cases in Karnataka, out of which we sequenced about 100. At this point, sequencing was not so, the sequencing methods were still being developed. So this was, and we had access to about 369 samples. And so roughly, these are the different districts of Karnataka. They've been sort of, the heat map is for the number of cases that were there at that time. As you can see, this is Bangalore here, Bangalore urban. And basically, we got a lot of samples from this region and we sequenced a lot from this, but we also sequenced from other places. So those that were sequenced are in lime, and what were tested, what was available to us was in dark green. Right, and we had information on all of these people. So we had their age, we had we sort of knew who they were in the sense of male, female, clinical state, nature of exposure, and we worked with the government agencies to work on this data. And we had the travel histories. We knew whether they were community, like cases that had come in without any known contact and so on. And what we did was, after sequencing, we built this sort of network of looking. So each sort of circle in this network is a person, and then their known contacts are linked. What it does not have is all the people they came into contact with. These are only positives, right? And so you see multiple clusters. We have looked at, so all these are clusters of cases. So they are based on actual tracing and figuring out who was in contact with whom. And so these have been colored by the lineage of SARS-CoV-2. So we think of A and B. So these are pangolin lineages. What that means, in that nomenclature, A and B are considered the ancestral lineages of SARS-CoV-2, sort of in that was circulating in the market in Wuhan. And after that, all subsequent lineages will carry, for example, B.1 is a daughter of B, and B.1.1 is a daughter of B.1. So basically, from the lineage, you can know the ancestry. That is how this phylogenetic nomenclature system is designed. And obviously, there are distinguishing mutations that separate B.1 from B, right? So that's just to give you some context on that. So at this time, we found that there were multiple lineages in Bangalore already. This is very early, in the state of Karnataka already. And there were also continued introductions, right? So we had for example, this A was from travelers who had come from another state. So basically what we were able to say really early on, that there were multiple introductions of SARS-CoV-2 into the state of Karnataka. There was continued introduction even during lockdowns. So there was some porosity in state borders and so on. And we looked at the phylogenetics of this, and we knew that they were quite distinct. We also knew which were the sort of cases that were not falling within clusters, suggesting that this virus was in community, right? So these were known, they were contacts, but they were not, I think they can, they're contacts. And we also then looked at how many of these were asymptomatic, and we sort of had this idea early on that it looked like people who were symptomatic seemed to have more number of contacts who were positive. So the middle of the clusters with the people with the most number of nodes as, is that what you call them? The number of connecting, are these edges? Yeah, okay. The number of connections seem to be the people who were symptomatic, as well as, both the starters of the cluster, which we had some indication on, as well as the people with the most number of contacts seem to be the symptomatic ones. But we didn't do the statistics on this. These were small numbers. And we also looked at how these were localized in terms of the districts of Karnataka, and we found that most of them were in single districts. So these are colored by districts, and you can see that most of the clusters are in single districts. One more thing that I should mention about here is that we didn't sequence everyone, but you can see that in most clusters there is a single color, right? That means it's suggestive of the fact that that cluster had that lineage, basically. You can see this one large cluster with two colors. We don't know whether this mutation was acquired independently or there were two distinct introductions. This clearly is a case where this person was unlikely to be linked to this cluster. So we got these kinds of information, and we were able to feed back on this, right? Right. So some of the clusters were well contained within districts. Some of the clusters had spread to multiple districts. That is sort of, and the largest cluster obviously had spread to the nearby districts. So this was the first piece of work that we did. We identified multiple introductions. Most of the large clusters comprised of single lineage and were restricted to a few districts. We were able to assign lineage to cases with no known history of travel or contact. And so we were able to get a sense of whether, which lineages were not being contained, right? And we could get some ideas about that, about what was happening in that outbreak. Continued importation by domestic travel, we could see, because we knew the travel information for these people, we actually were able to get some ideas about that. Symptomatic individuals. So at that time, it was not clear if there were some lineages that tended to have more symptomatic individuals. From our data, the limited data that we had at that time, it looked like symptomatic individuals are spread across multiple lineages along with asymptomatic individuals. So we didn't see a pattern there. This was one of the first studies which linked epidemiological data with genomic data, and we thought that it was important to do both for effective outbreak management. So moving on from then, towards the end of that year, we started looking for variants. So this was in travelers. So we got, so when people were coming back from the UK, for instance, we got those samples and we were checking them. So this was late December 2020, and pretty early on, the VOC classification happened on 18 December. And in a sample that was collected on 22nd December, we detected alpha, and we also put this up on GISAID by the end of December. And so we were able to detect a variant that was circulating quite early, and this contributed to an understanding of how fast things were spreading to different countries, right? So that was an important contribution of that. We became good by this time at sequencing from travel-related cases. We could sequence from outbreaks. We could recognize that VOCs were spreading, variants of concern were spreading rapidly, but it wasn't clear if we had a VOC that emerged locally, whether we would be able to detect it. And this was the time, so this is roughly, we are looking at in this time, so the sequencing window, between the first and the second wave, right? So that's when we are looking at that window, we could see the emergence of something coming up reasonably early if we looked at the data, but it took us a long time to get confident that there was a variant of concern here. So this is the Delta wave. So first you see Kappa and then Delta in Karnataka. This is only Karnataka. Initially, very early on, but if we did, the expectation was that you start seeing. We had started seeing clusters of cases towards the end of Feb, but it was Kappa and not Delta at that time. So B.1.6, 17.1, and but we were not able to pick it up as a variant of interest or variant of concern because our sequencing was very limited at that time. Right. So that was yeah, we could have done a bit better at that time. And after that, of course, INSACOG was set up, did way more sequencing, ramped up sequencing, and we were able to identify things a bit faster. So at this point, it was clear that we needed to scale the sequencing efforts. And INSACOG had already been set up by that time. The labs were getting recruited and people were getting trained to do this. And we had to track mutations and viral evolution over time and analyze sequence data in its epidemiological context, but at a large scale. So then I joined Strand Life Sciences, where we did more than 5,000 genomes between July to December 2021. And we also started reporting this to the municipal authorities. So for the first time, there was genomic information that went in a sort of, the idea was, can you interpret this data? And actually, even though sometimes it wasn't clear what you would do with it, right? The report still was something that went out. So we standardized that process. And then we built an analysis platform for a large number of sequences, combining genomic and epidemiological information. Now, what we do at IDRF is we are doing hyperlocal surveillance. The idea is that there might be early warning for this and also identify variants early. So we also want to sequence from positive samples. This is 15 sites across Bangalore, including the international airport. We have some hospitals, we have some apartment complexes and some institutions as well. So as you can see, this is from we started work from middle of June and till December now. And for all these, each color is like one site. So it gives you some rough indication that we saw SARS-CoV-2 in wastewater in June, July, and then we again saw something in August and September. But since last 4-5 weeks, we haven't seen anything in wastewater. No, we know that they correlate with cases as well. And right now, yeah. False. Oh, so I mean, there were cases in the community at that time. I guess that's what they tell you. And because we got this baseline only now, like we will be able to tell if something takes off. We didn't have this before for our data set. Right. So I think looking back, I think this is a sort of framework that I think is important here. We had to build the tools. We had no sequencing was not so mainstream before. There were very few labs in the country who could do this kind of sequencing, even amplicon sequencing, right? And so the sequencing protocols, even though we did take protocols that were existing around the world, it worked for labs across the world. Like that is among, like, it is challenging to build that kind of protocol, right? And so we got some experience in doing that. So now to sort of do this for other viruses becomes easier. We had to make the informed decision on whom to sequence and when, right? And we got a lot of experience in this as well. That's what the initial part of the pandemic helped us do. We tested these tools. When we had the variant of concern coming in, we were able to generate this information in a timely manner and report it. And because we had feedback from that, we could build the confidence that we are doing this right. And it's very hard when you're working in isolation to actually know whether you're doing the right thing or not. And I think that if we don't do this enough number of times, we'll never build this confidence. So this testing of our abilities was important for that. We achieved some scale, both from INSACOG and from Strand. And this model was already there in the world that we need to do scale. And that came mainly from the UK COG model, right? So they had already shown that there is some value in doing scale, because otherwise what is the value of sequencing thousands of genomes? That wasn't an established thing before the pandemic. And when you do scale, I think there is a challenge that you might lose sight of what is important. And I think that depends from project to project. So in this case, we should keep track of what are the mutations that are coming up, not just put 10,000 sequences online, right? So, and sustainable solutions, maybe it isn't sustainable to do look at human cases of SARS-CoV-2 over time in the kind of depth we want to. So when there is no crisis, when there is no outbreak, maybe something like environmental surveillance is a better way, but we need to continue doing it. We can't stop and then start at another time. So those are sort of the frameworks, and I would love to have other people's thoughts on this. This is the other thing that I sort of work on that I'll quickly talk about, which is basically, are there undiscovered, underappreciated pathogens already causing disease in humans? So basically infections of unknown etiology. Mainly I work on fevers and mostly brain infections. So brain infections, there is encephalitis, meningitis, and meningoencephalitis. Basically, there are many pathogens that can cause the spectrum of disease. And they include viruses such as Japanese encephalitis virus, dengue, chikungunya, Nipah, etc. Bacteria such as Orientia susugamushi, which causes scrub typhus, Streptococcus pneumoniae, and many others, and multiple fungi and parasites. So because the pathogen spectrum is so large, there are many, first of all, because of that, and also because we don't know what is circulating. Sometimes we can't tell what the person has. And so, yeah, this is just to give you an idea of what has been reported in India. So so you will see among this acute encephalitis syndrome, acute diarrheal syndrome, and so on. So there was a study already in place. It was a 4-year enhanced surveillance study that was set up in multiple states in India. I think Assam, West Bengal, Uttar Pradesh. And I sort of piggybacked on this study to do some sequencing work. So here the samples were already available. People had already done most of the work. And I was looking, so what I do is I use nanopore sequencing. So in this sort of method, you have a pore through which DNA passes through. The electrical signal generated tells you what sequence it is. I can talk more about this, but that's roughly what it is. It has been used in extreme environments. So we have taken it to UP and we have taken it to Assam during outbreaks. And I'm going to tell you a little bit about one virus that we discovered, which is not commonly, actually it's not, we didn't discover it. We found it associated with cases of meningoencephalitis in West Bengal. So this is a erythroparvovirus 1. It's a small virus. It's known to circulate in India. It has been found to be associated with cases of meningoencephalitis in the UK, but we don't know whether it's causal. We don't know. So people have detected the presence of this virus in these cases, but a lot of work needs to be done to say that this virus is causing this disease. Its transmission is mainly by respiratory secretions as well as contaminated blood products. So basically, we had access to 403 samples. We tested them with PCR for B19. We got 13 of them to be positive. They didn't cluster in any particular way, but some of them had other etiologies. That is, they already had a Japanese encephalitis virus also in them. Some of them had other pathogens, but 8 of them were unknown etiology. Okay, right. So those are the things that I wanted to focus on. What we found was, so basically in 13 out of 403 samples from meningoencephalitis cases in West Bengal, we detected B19V genomes in them. Many of the positive samples were from children. B19V was identified with other pathogens. It's the first report of B19V from meningoencephalitis in India. We sequenced a complete genome from cerebrospinal fluid. So like I said, we had that expertise now from which we could then translate to different viruses. So this belonged to a distinct clade, genotype 3b, which had sequences from Brazil and Ghana. And most of the positive cases actually had good immediate outcomes. 10 of 13 recovered and were discharged. 2 left against medical advice and 1 person died. That person was also positive for JE. And when we looked at this, so this is genotype 3b. So this is the phylogenetic tree, what we're looking at. And so these are the different genotypes of B19V parvovirus. And this is genotype 3b. This is where our samples sit. If you sort of blow that up, you see that it sits. These are the only sequences from cerebrospinal fluid. All the others are from bone and blood and so on. And we also looked at partial sequences because partial sequences are available from India and they cluster with sequences from India. Right, so we don't know whether this is causal or not. Yeah, so this is just a sort of summarization of that. So I just wanted to show you two different things, one about data and one about samples, and how having a good surveillance network and then the collaboration to have both the data as well as work in the wet lab, it has allowed us to uncover these stories. That's it.
You showed these different lineages early on that you were tracking. Can one fit a probabilistic model to how lineages evolve and then try and say something about, okay, what's the probability that something dangerous will occur? Is that kind of effort viable?
So initially when we saw the SARS-CoV-2 genome, we thought that the spike wouldn't mutate that much. So there were assumptions that we took on. But it was very clear that there is a lot of diversity in spike. So can we predict what's going to happen in spike in a certain sense? I mean, I think it can be done, but it seems to be way more, the virus genome seems to be way more plastic than we would have anticipated. But there must be states that the virus can't be in and states that it can be in. But it hasn't. But also because mutations are additive, right? So then I wonder if the number of combinations we're looking at is really huge. It's 30,000 base pairs long. So, yeah.
No, I'm just wondering if now one can say that, okay, chance of a new dangerous mutation coming out of China or somewhere is of the order of 10%, 20%. Can one begin to make comments like this? Yeah.
I think it's worth thinking about it. I think we can. Yeah, I think there's enough genomic information to ask this question about what sticks and what goes away. But that depends. Like the virus that is fit in the lab versus the virus that's fit in the world seems to be so different. And even if you look at different places, right? So we had this sort of expansion of B.6. No other place, I think only one other country in the world saw that kind of expansion because very quickly B.1 took over everywhere. So it depends, it seems to depend a lot on the local epidemiological context and then super spreader events, like a lot of things, right? So I don't know.
And was Delta in this genomic sense, was it surprising or was it just one more thing that showed up?
So actually, when we initially found Delta, we could see that there was a constellation of mutations that we were seeing again and again. The confidence to call it a variant, we acquired later. Because what we want is you have a constellation of mutations, you see it again and again, you see it increase, and then you also see cases increasing. So you want all of those to fall in place, and to collect all that information took time.
Any other questions?
This is related to the previous question. Given 1,000 people, a population of 1,000 or so, with one strain, what's the actual data on approximately the number of variants of concern that will emerge and what will be the time scale? Some numbers are available?
So I guess we didn't look at it in that way, but with this data we can. So yeah, so how many of those were introductions versus how many of them were local evolution? Right. The only place where we saw sort of local evolution was maybe in that one large cluster. Everywhere else it was just a single kind of virus. But when I say single kind of virus, we still didn't look at the mutations carefully enough. So this is something that wasn't done, but we could look at that.
Hey, so I had a question about mutations again. So when are these mutations actually introduced? During the time that the virus is replicating in a host, or is it when the time that it's transmitting?
I think it's during replication.
Replication,
yeah,
okay.
For an RNA.
Yeah. And then basically you are saying that you need to see enough number of mutations, and maybe in a phylogenetic analysis it has to be on its separate clade using certain statistical or bootstrap analysis to call it a variant. Is that how you will declare a new variant?
Also, I think for when you say it's a variant of concern or variant of interest, now we have a sort of checklist of things. Either it should be associated with an increase in cases, or there should be increase in reinfections or vaccine escape or serious disease, hospitalizations. So multiple of these things will be looked at the same time.
So even for de novo variants, you will apply the same criteria and then identify that a new variant has emerged.
Sorry, that is just for variants of concern or variants of interest that
Okay.
are being tracked in some kind, like where everyone is now tracking those variants. Just to call something a variant, yes, it just needs to belong to a different subclade. And
Okay.
then what is that? Actually, what people do is they raise an issue in pangolin, and then someone looks at that and says, okay, this is different enough. So, and assigns it a new name.
Okay, so my final question is, like slowly evolving bacteria like TB, for instance, there are cases of mixed infection where different strains could coexist within the same individual. Here, this is the time is so short, but have you seen any cases of mixed infection in COVID as well, different lineages or strains in the same
Yeah.
individual?
Yes, people have reported this. Also, some of the variants that are circulating now, they are recombinants, which one of the ways that can arise is if a person was infected with two viruses at the same time.
Oh, I see. So, oh, thanks.
Any other questions? If not, let's thank Dr. Chitra once again.